I'm Adriana Norris. I'm a member of the ASBMB Science Outreach and Communication Committee, and today I'm so excited to be interviewing Dr. Joshua Sokoloski, who was awarded the ASBMB Science Outreach and Communication Grant. So this award supports ASBMB members in organizing community engagement activities and trying to foster an appreciation for science in informal settings. So Dr. Sokoloski is an assistant professor of chemistry at Salisbury University. He is a biochemist that is focused on science outreach and education and all things related to RNA and DNA. So welcome, Dr. Sokoloski, and thank you for being here.
Oh, thank you for interviewing me. This is going to be great.
Yeah, I'm excited. So today we're here to know more about Dr. Sokoloski's mission and interests, to learn from his creative process in organizing a successful outreach program. So we can go ahead and get the interview started. So the first question I wanted to ask is, why is science outreach important to you?
Well, I view outreach as an intrinsic part of my job as being a scientist. We learn new things in science and we tell other people about those new things, but we can't just tell other scientists, we have to tell everyone. And I think it's important for science to be open and accessible to everyone, whether they're in the field or not. And the best way to do that is get scientists themselves out there to talk about their work.
Yeah, definitely. I totally agree. So can you describe your Decoding the Mysteries of DNA program that you did?
Sure, yeah. So this was part of the summer Enrichment Academies that we hold at Salisbury University. And we had a week-long summer camp focusing on STEM. And my program was a 2-day part of that. But eventually we had a 3rd day at it. And we can talk more about that later. And the first day activity was focused on learning about DNA, the history of DNA research and discovery, the structure and function relationships of DNA. Now, for a middle school audience, of course, so we kept it very basic. And, we learned about base pairing, and we learned about base pairing using magnetic 3D printed molecules. And the students could figure out, oh, these two don't match, but these do, and they click together. And so it's a way for them to explore and figure out base pairing in a hands-on activity. We also did some DNA extraction from different types of fruit, and we had to hypothesize which type of fruit gave the best. Turns out that strawberries were the best, with blueberries a close second. And we also learned about the genetic code and how to decode a genetic message. And the second day was later in the week, and DNA was part of the forensic science activity we did that day. We had to solve a crime. And the theme really was fingerprinting. So we did fingerprinting in, of course, the human sense, fingerprinting in the DNA sense, and looking at DNA from a crime scene, and also chemical fingerprinting, taking chemical residue from the crime scene and using chromatography to separate out the components. And the 3rd day was sort of just a fun day. It was the end of the summer camp. So we did fire and ice type activities. We set things on fire, we blew things up. We froze lots of things with liquid nitrogen, and we made liquid nitrogen ice cream at the end of the day to conclude the week.
Yeah, that all sounds so fun. So what was your mission for the program?
Well, the primary goal was to teach DNA structure-function relationships in a fun and entertaining way to a middle school audience. Hopefully teach the students a little bit about DNA and RNA, and also what biochemists do as a career, and what do we do in the lab, and what's it like being a scientist.
Yeah, that's awesome. Do you feel like the mission of your program was achieved?
Well, I do feel that they definitely learned more about what a base pair was leaving the room. And they certainly learned how to use a micropipette and run a gel and do some real science lab activities. And I hope, I can't be certain, but I hope at least some of them have a little bit of excitement about biochemistry now. And maybe as they go on in high school and later on in college and beyond, they start to take a more focused interest on those biochemistry topics.
Gotcha. Yeah, you fit a lot into 3 days. That's amazing.
Yeah, it was 2 sessions a day. So I did 1 session in the morning, and then I repeated it for the second session in the afternoon. there was 40 some students, and we split them into two groups. And so, yeah, it was a lot of students. So
yeah, that's great. So how did you advertise this program to your community to get people to kind of come and engage?
Well, it was great to get connected to the Summer Enrichment Academy because they took care of all the advertising. They did social media advertising, and they did a lot of mail and email campaigns to local schools. And, we have flyers that we gave to local schools too for it. And the Summer Enrichment Academy director, Wynette Curtis, she did a lot of the advertising and outreach, and we got a full roster of signups by the end of February. So it was actually pretty good.
Yeah, that's great. Yeah, it seems social media is definitely a good outlet to get to talk to the community for sure. The final session of your program was kind of impromptu because another session, got canceled. So how did you overcome this challenge of sort of planning this last sort of impromptu session?
Well, I do a lot of outreach, and so I have a lot of activities sort of ready to go. And we have a lot of the stuff. Our department has a whole room dedicated to demonstrations and outreach activities, both for in our classrooms and for outside events. And so I really just went through the room, looked to see what was available and set up. And I just chose things I did before and I knew were crowd pleasers, and I just put it together. It really wasn't that stressful. That's why I volunteered to do the third extra day, because I knew I could just walk 10 feet outside my office and find all the materials I needed to go do some explosions and liquid nitrogen ice cream. So.
Yeah, that's awesome. It seems like when you do a lot of outreach, you sort of have these things that you can sort of repeat and already have the materials. That's awesome.
Yeah, and once you do it the first time, like these models, now they're here forever. I can use them for classrooms and other outreach activities.
Yeah, definitely. So did you overcome any other challenges during the workshop? And if so, how did you overcome them?
Yeah, I think it was pretty smooth. The biggest challenge outside of this extra day was not quite knowing the full roster of kids until Monday morning, because the parents had to return permission slips and all that, and so I had to aim for the maximum amount, and it was good I did, because we did have the maximum amount, but we weren't quite sure the numbers until the day of. So yeah, a little bit of flexibility there. So always plan for the maximum possible, and if you get less, that's okay. But don't plan for less. Always plan for more.
Yeah, that seems like really good advice. You mentioned too that the audience for the Decoding the Mysteries of DNA program is mostly middle school students. So how and why did you choose this particular audience for this kind of programming?
Well, traditionally, a lot of our STEM summer programming have target middle school students, because it's a good age to try to engage them in science and get them interested before they get to high school, and they can start taking some of these science courses, maybe take some more advanced courses, take some AP chemistry, AP biology down the road. So it's a good age to get them started down the road to a science career if they're interested in it. And also, there's challenges with this age group too, right? And the key thing is to keep them engaged at all times. I can't talk like I would in a college student lecture for 30 minutes. I have to break it up with hands-on activities. And, really you gotta, as a leader of an outreach, you have to be ready to transition into another activity as soon as you start seeing your audience attention start to wane. So you really have to pay attention to the temperature of the room and figure out when it's time to move on to the next thing.
Gotcha. Yeah, so you have to be engaging, but also flexible to move on to something when you see your audience sort of, getting a little bored or falling asleep.
Yeah. And that goes into the planning too, is to make sure that everything's set up and ready to go, so you can just go to the next activity as soon as you want to.
Yeah. Yeah, that seems really smart for this kind of audience. So are there other audiences you would like to reach in the future with this workshop?
Yeah, I would like to do sort of a high school age workshop for juniors and seniors in high school, and maybe have a whole week long of DNA structure function experiments. Have them do set a hypothesis on day one and do some experiments and see the results by the end of the week and really guide them through a real science experiment. And for that to work, I think a little more maturity in older students would work well, and sort of get them ready for, college and maybe undergraduate research as well.
Yeah, that's awesome. So you also mentioned that you had several collaborators, including from the Summer Enrichment Academies. So how did you go about sort of building these collaborations?
Well, the connection through Summer Enrichment Academies came through Dr. Mark Moeller, who was the Associate Dean of the Henson School of Science Technology at the time. And so he put me in contact with the right people, and it was great to have him because he knew who to talk to, and we set it all up. And then I showed them my program idea, and they approved it, and we included it all in there, and it worked great. And so he really, made sure everything went smoothly. The logistics were handled quite well. So it's always nice to have people in your corner to help you prepare for that, and people who have the power and experience to be able to make these things go smoothly as well. For the 3D printing, this help was done with Chris Woodall over in our libraries, and he helped me design and print these molecules and handle troubleshooting. There's always some troubleshooting when you start printing molecules. For my student helpers, I went to the ASBMB student chapter meeting. I presented my ideas. I got several volunteers, but because it was summer, only one of them were able to come and help me, Emily Hopkins, who's a student here at Salisbury University. And she did great. She helped me present some of the topics, and she was in charge of showing students how to use a micropipetter to load an agarose gel, and she did fantastic with that. It worked really well. And also the Summer Enrichment Academy provided us with camp counselors. So these were SU students who were education majors, and three of them I had throughout the week was Skylar Ramos, Susanna Randall, and Lindsay Wigatow. And they were fantastic with helping keep the room well behaved. If there's any behavioral issues that come up, and sometimes it is with middle school students, they were right there to make sure there was no safety hazards going on. And it freed up me and Emily to do the actual science teaching, and any taking students aside to tell them to adjust their behavior was done by the camp counselor. So that was fantastic. That was great.
Yeah, that's awesome. So in your other outreach projects, do you usually have to do sort of all those roles, like sort of like a disciplinary, and is that sort of difficult, or do you usually have people helping you in that way?
No, usually it's just me by myself. And I've done this before with the Boy Scouts, merit badge colleges, and sometimes it's hard to guide the scouts through the activities and also address behavioral concerns, because it's around the same age group you have in there too. So having someone in there who was a camp counselor to help with all that was fantastic, and help get the students from my room over to lunch, or back from lunch over to my room for the afternoon session. That was great too.
Yeah, it seems important to have those collaborators then.
Mm-hmm.
So it seems like the Decoding the Mysteries of DNA workshop was a really big success. Are there ways that it could be improved, you think, though?
Yeah, everything can be fine-tuned. Like all these activities I've done multiple times before, and every time I do it again, I change something to make it a little bit better. For instance, I mentioned, and we showed a little video clip about Jurassic Park, the 1993 original movie, about DNA and how DNA, encodes for living creatures. And the students really liked that. And one of our best discussions we had all week was talking about cloning different animals and how that works, and why we should do it or why we shouldn't do it, and having that discussion. So they were really engaged with that. And so the next time I'll run this program, I'll definitely have more about Jurassic Park and cloning in there, because the students this time really liked that aspect. And so I'll make sure it's in there more the next time.
Gotcha. When you ask them if you why you maybe shouldn't clone something, are they like because they might eat you?
No. Some of them were worried about if cloning could cause disease in the animal coming out, and I had to go through and say we're not really cloning dinosaurs. We can't, because the DNA half-life simply won't allow for intact DNA to be recoverable from dinosaur samples. But the projects out there now to clone animals are always animals that were extinct through human problems, humans causing them to go extinct. So it was a way to undo human past mistakes in the ecosystem. And that would be the primary reason now why we would want to clone an extinct animal and bring them back to life.
Okay, yeah, gotcha. So have your outreach efforts influenced your career trajectory? And if so, how?
Well, doing all this outreach is so much fun that I've definitely shifted my scholarship here to focus not just on discovery-based research with RNA and DNA chemistry, but also to come up with innovative new ways to teach things in the classroom, in the laboratory, and to do things, in outreach activities. So I put a lot of my scholarship time into developing these activities and carrying them out. And also it's always a good thing to do for a career to put yourself out there and get known with the community, and being the person who's willing to go and be the face of science at SU is always a good thing.
Yeah, that's awesome. So it seems like you're sort of an expert on outreach projects, and you've done it a lot. Can you sort of share some advice for other scientists who want to get involved in an outreach project or even create their own, of sort of how to start doing that?
Yeah, just treat it the same way you would do your research or you would do preparing for teaching. So, you want to practice and prepare, refine, revise, always pay attention to your audience and see what's working and what's not working, and make notes and change for next time. So you're getting things better each time you do it. But I think another key feature is to always have fun when you're doing it. So whatever you choose to do for outreach, make sure it's something that you enjoy doing yourself, because enthusiasm and excitement is very contagious, and it's good for students and people in the community to see that you really love what you're doing, and that's why you chose the career that you're in. And that's always a good feeling too, to show that there are people out there who really love science and they're doing it for their career. So maybe these students, if they really love science, they can realize they can do it in a career too.
Yeah, that's awesome advice. So thank you for telling us all about the Decoding the Mysteries of DNA program, and also giving us that really helpful advice on how to get involved in outreach. So thank you for the viewers for watching, and we will see you next time.
Oh, thank you. Yeah, bye.